Then I'll pause the recording for the secret question. Yes, Maya, actually, you can find the answer to all of the secret questions that have been asked.
Yo la tienen a las 8, amor.
Sorry, I don't know who has their mic unmuted, but yes, Maya, you can find the answer to the recent secret questions in the About Your Instructor and Assistants tab right here, actually. So look there, and you'll find all the answers. And then I think Kimia had their hand raised. There's also another question in the chat that I can't even, it's not letting me scroll.
Sorry, I don't know how to put my hand down.
That's no problem. I got you. Actually, no, you got you.
Yeah, I got it. Thank you.
No problem. So hello, everyone. Welcome. I should actually join that.
So the first thing that we'll do is go over quiz 6 because there were quite a few issues and like confusion with that stuff. So we want to go over that before the next exam. And then after that, we can go over whatever you guys want to go over. And then Garrett, do you want to do the secret question now?
Oh, yes. Yes. 1 second. Let me pause the key. There we go. So, do we have any questions to start with?
Quiz 6, Garrett.
Oh, yes, quiz 6. Okay. Actually, let's put that away. Let's see. Quiz 6. So, Dr. Chiari, Sam, and I had been looking through these, and we weren't really a fan of some of the answers we were seeing. Just on the whole, a lot of people did a good job, but not everyone did. So let's talk about it. So in a population, you have the following individuals. These guys. What is the ploidy of this organism? How many loci are shown for the individuals above, and how many alleles are present for each locus? Okay, did you want to do this one, Sam?
Sure, I can go ahead and do this one.
Okay.
Okay, so can anybody point us in the right direction of how to begin answering this question? Because sometimes it's easier to hear it from peers than it is from us. Yeah, we'll start with ploidy. Okay, yes, so everyone is right. It's a triploid because, yes, so Maya is right. Maya said that isn't the number of copies of an allele what determines ploidy? And yes, so we can see in, say, like the first individual or the first 10 individuals, there's A1, A2, and little a. And so there's 3 copies of that allele. And same thing with our B's, C's, D's, and E's. And the same thing goes for our second one, the second line and the third line. So that is how we're going to find our ploidy. And so this group would be, or this organism would be a triploid. Yeah, and Usman, you can definitely just look at the last one and also figure out what the ploidy of the organism is. And so the first one was kind of tricky because it had all the numbers, but as long as you're looking at as long as you can find it for like one individual or like one of the genotypes, and then it's consistent with the other two genotypes, then that's completely okay. All right, and so now that we know how to find the ploidy, does anybody want to take a stab at finding how many loci are shown? Yes, so Ori is correct. Loci is found with a number of different genes, and then Joseph said that the loci is just how many different letters, and that's correct. So in this, for all of these individuals, we have 5 different genes or 5 different letters. So we have A, B, C, D, and E. And so that's just all 5 of our genes that we have. And so we're only looking at 5 loci since we have 5 genes spread across 5 different locations on our chromosome. And so finding the loci is as simple as that. You just have to find how many genes, or in this case, letters that you're dealing with. And then for the last question, can anybody tell me how many alleles are present at each locus? Do you think it's 3 for all of them or some of them? All right, so Joseph said 3 for some. And Maya asked, is it how many different forms for each type? Yes. Okay, so oh wait, Joseph is saying he thinks it's 3 for all now. Okay, so Maya said sorry, we're kind of going everywhere. I'm just reading off what the chat says right now. So Maya said that for E, she thinks it's just going to be 1 allele at that locus. And yes, for E, that would be correct because we only see 1 kind of allele present, and that's big E. Like that's literally all we see. So for E, we're going to have 1 allele present at that locus. And then if we look at, so I guess we'll jump back to A, sorry. So for A, we can see that we have a big A, and then we have A1, A2, and then a little a. And so because we have big A, or we have A1, A2, and little a, that's just 3 different alleles at that locus. So there'd only be 3 alleles located at the A locus. And I know that that can be kind of confusing. And at first glance, you go, oh, okay, so there's 4, but I think, and Garrett, correct me if I'm wrong, but for the second genotype, that big A is the same thing as just like A1 and A2, right?
Let me check really fast. I wouldn't originally think so, but
I didn't think so either, but when Dr. Chiari sent us
yeah, that's what I was.
the answers to this question earlier today, she said it was only 3 at the allele, and so that was my reasoning behind it.
Yeah, I would think that because we have A1, A2, A, and A, that it would be 4, but
Yeah, that's what I thought too. So I
I'm
was
not sure.
a little bit of confusion, but I was like, Dr. Chiari knows all. So
So you could argue that it's 4, and I would support that argument.
yes. So sorry, that's kind of inconclusive about A. Yeah, Nick, you might be right. I think it might be A1. I'm not sure, but why did Dr. Chiari say 3? So Honestly, I'm not entirely sure why she said 3. Because when I first looked at this question, because someone sent it to me before she sent Garrett and I the answer to it. And I also thought it was 4 for A because you have A1, A2, just a normal A and then a little a. So unfortunately, I could not tell you why it's 3 instead of 4. I'm sorry that I cannot answer that question. I wish I could. So the number of alleles present at the A locus is 3. And, I'll email Dr. Chiari after this and ask her why it's 3, because I would also like some kind of explanation. But moving on from A, we can look at our B locus now, which is much clearer and straightforward. So we can see we have B1, B2, and little b. And in the second one, we only have B1s. And in the third one, we have little b's. So we are only looking at 3 different kinds of alleles. We're looking at B1, B2, and little b. So for the B locus, it is 100% going to be 3 alleles present at the B locus. And then we can go ahead and look at C. And for C, we have C1 and little c in the first genotype. And then in the second one, we have C2 and little c. And then in the last one, we have little c. And so for the C's, we're only seeing C1, C2, and little c. So that means, again, yes, we will only have 3 alleles present at that locus. And then we can look at D, and we'll have 3 alleles again because we have D1, D2, and little d. But Garrett, when she sent us the answer for this one, she said it was 2.
Yeah, I think there's some maybe inconsistency.
It's all right. We're going to say it's 3. And we will double check and let you know.
I agree.
And that is how you'd walk through this question for ploidy. How do we feel about it now? Are we less confused? I hope less confused. Oh, Joseph said this makes no sense. Okay, so for each locus, so Joseph, yes. So for each locus, we're only counting the different alleles. So for that third question, we're being asked how many alleles are present for each locus. So one locus represents 1 gene. And then that means that we are just going to be looking at 1 letter for each locus to find how many alleles there are. And to find how many alleles, you just look at how many different ways the letter is written. So like for our B's, we had B1, B2, and little b. And so that's like 3 different ways that B was written. Okay, so Joseph said he put 3 for all of them because there are 3 alleles at each locus. Yes, there are technically 3 alleles at each locus, but for example, for E, they're not 3 different alleles. They're all the same allele. So there's only 1 kind of allele present at each locus.
Yeah,
I think
right.
a lot of this comes from like just doing practice problems. And so when you do a lot more practice problems, and like even with these quizzes, you start to understand like how questions are asked and how they might be presented and what kind of answers it's like trying to pull out of you. Casey said, but the question just asked for the alleles, not different alleles. So, and then just said, I make no assumptions. Okay, so yes, it did just ask for alleles, but it wanted to know how many alleles were present at each locus. So I do see how that could be confusing.
So you are right that 3 alleles represent the gene at every locus, but if we remember what an allele is in the first place, an allele is a specific variant of a gene. We see that there's one variant, two variants, three variants. So yes, three alleles do represent the D gene at the D locus, but in the case of these three individuals, there is one, two different alleles. But yeah, the wording definitely could have been more clear.
Wait, so what's the final answer? Are you adding up all of them, like locus A, B, C, and D, E, and then putting the answer?
For each locus, at locus A, there are 4. At B, there are 3. C is 3. D, 3. And E is 1.
Oh, that's the answer?
Yes.
Oh, okay. Yeah, thank you.
You're welcome. I do think you got some partial credit for that if you just said 3, because, at the A locus, B locus, C locus, 3 individual alleles do represent that gene, but that's not what Dr. Chiari was really going for.
Yeah, Joseph, for D, saying that there were 2 for D, we're gonna check on that and see why. Yeah. Yeah, there were many different questions in the question pool, so everyone might have gotten different questions. Yes, Casey.
So just so I understand clearly, like if we were to just look at the first genotype consisting of 10 individuals, if the question asked how many alleles are present for each locus, it would be for A, it would be 3 because you have A1, A2, and little a, and for B, it would be 3. For
Mhm.
C, it would be 2.
Yes,
For D, it would be 3, and for E, it would be 1.
yes,
Because
exactly.
essentially, like, because it's an allele, we're looking at like big E, big E, big E, they're all the same. So they're not really alleles because they're not variations of the gene. They're all the same
Yes,
gene because it's all dominant.
yes, exactly.
Okay. And then to find the loci, we just look at all like, okay, we have 3 A's, 3 B's, 3 C's, 3 D's. So it's 3. How many loci are shown for the each? So that
So,
would be, it would be like A, B, C, D, E. So it's 5, right?
yes, exactly.
Got it. And then for ploidy, it's kind of similar, but not because you have, it's triploidy because you have 3 different letters across the board.
Mhm. Yes.
So the ploidy of the organism is informed by the number of alleles that represent the gene at each locus. So because we see 1, 2, 3 A's in a single individual, so the ploidy is kind of like in humans, you have your diploid, because let's say at this A locus, a human has 1, 2 alleles that represent the A gene at the A locus. That's what makes them diploid. Di means 2 because there are 1, 2 alleles. In these individuals, these organisms, there is 1, 2, 3 alleles that represent the A gene at the A locus, and therefore it is triploid.
Okay, got it. Thank you.
You're welcome. Oh, and someone had a question earlier that I wanted to respond to.
Garrett, while you're looking for that question, Caroline, yes, for ploidy, you can write it as 3N because that's the same thing as being triploid.
And yes, Joseph. And yeah, I'm sorry. It's a bummer that it didn't articulate that it wanted to know the different variants that we see, but you know.
But in the future, the likelihood that you'll probably see this question again on the exam is probably high, because I think a lot of people are confused about it. And so she'll probably want to make sure that you understand it now. So yes, in the future, assume different.
And if you want to explore it some more, very cool. Never mind. If you want to explore it some more, you can find 2 questions pertaining to this kind of question in the week 7 review. Now let's move on to the next one. It's more of the same. Aita says, for triploid, how many alleles would you say there are? Well, a triploid individual, for example, having an A genotype, how many alleles? 1, 2, 3 alleles represent the gene at this locus. That's what makes them 3N, 1, 2, 3N in the first place. But in this case, there are 2 different variations of this gene, 2 different allele types, the capital and lowercase a. And yes, Caroline. Okay, genotypic frequencies, someone asks of it. Okay, so for this, we add them all up and then divide by group, right? Like total is 18. So yes, we can add these up. 7 and 6 is 13 plus 5 is 18. So the population in this question is equal to 18 individuals. And so the genotype frequency, firstly, let's identify what are the genotypes of these individuals. What is the genotype of individual, or the first 5 individuals? This is important. Yes, exactly. This entire thing, where'd the pen go? Okay, this entire thing is the genotype that is exhibited by these 5 individuals. The genotype is like the sum of genetic information that an individual has that expresses what genes they have, which inform what phenotypes or traits they can display. So this is the genetic information we have for them, and therefore it is their genotype. This genotype is made up of 3 loci, and each locus is represented by 3 alleles. So what would their ploidy be? Triploid. And how many different alleles are there at the A, B, and C locus? Bingo. So they are 3N because of the A, B, and C loci. And each locus is made up of 1 variant, 1 kind of allele, that being capital A in the case of A, capital B in the case of B, and capital C in the case of C. So what are the genotypic frequencies now that we know that these are our genotypes? It's just like Carla said, the genotype frequency of this genotype here is merely the number of individuals that have that phenotype, or excuse me, genotype, divided by the total number of individuals with a genotype, which is, the 18 individuals. So 5 divided by 18 for the first genotype, 6 divided by 18 for the second, and 7 divided by 18 for the third. And that is the very simple answer to this question.
I have a quick question.
What's it?
For the alleles present for each locus, you said one or two?
The
Or was that
alleles
not the
present.
question you asked?
Oh, sorry, what's that?
Or was that not the question you asked? Did you guys say what how many alleles present at each locus or no?
Well, this question doesn't ask that as part of the answer, but I was just kind of like extending based on the last question. And if I had to ask it again, I would ask how many alleles represent the locus or the loci for each of the individual genes, like the A gene, how many alleles represent that gene in this genotype?
It would be two. Because there's the capital A and lowercase a.
Yes, that's how many different alleles there are.
Okay. Okay.
Alleles present for each locus would vary with each genotype. Yes. In the first genotype, we see that there is only one kind of allele, one variant of the A gene, and that's being the capital A. But in this individual, there are two different alleles, two different variants of the A gene, one being capital A, one being lowercase a. But on the whole, this individual is triploid because three alleles, no matter what variant they are, represent that locus or represent the gene at that locus. Do we want to move on? Are we okay with that? Okay. What happened? You know, I can take the test again. Where'd it go? Oh, I think the time ran out. Let's find it. Okie dokie. Dun, dun, dun. So we got this one already. We got this one. Now we have this one. Okay. What browser? I think this is Microsoft Edge. Yes, okay. So, any ideas? Genotype. Oh wait, didn't we just have this question? What happened to the one that was, oh, dang it.
I think we did, but there's also like a pool of questions, so you might have gotten a different pool this time.
I thought I saw the genotype frequency question, or allele frequency, that's what I meant. But it like skipped it for some reason, and you can't backtrack, yeah. Okay, let's try it again. Shall we get this one? Okay, question 2. Oh, here it is, okay. All right, so question 2. What are the frequencies, not of the genotypes, but of each allele? Does anyone want to speak out and try their best? So before we answer this question, what is an example of an allele in or among these genotypes? Yes, there is the A, for example. There's also the A. There is B and B, as well as C. These are all of the different kinds of alleles that exist in this population. And so we need to find the frequency at which each of the different kinds of alleles exist within this population. And we can do that very easily. So we want to find the frequency of A, the frequency of A, of B. I'm going to spread these out actually. So we see that there's capital A and lowercase a, capital B and lowercase b, and only capital C. So those are all of the different kinds of alleles. And so how do we find the frequency of an allele in a population? Well, the frequency of an allele is, the number of times we see that allele divided by the total number of alleles in the population. Like if I wanted to find out how many green M&Ms are in 3 bags of M&Ms, then I would find all of the green M&Ms that exist within the 3 bags and divide that by the total number of M&Ms in all 3 of the bags. So for A, let's find out how many times when it's total number of alleles for the population, would that be 3? You're getting there. We aren't there yet. 3 times 5 plus 6 for total. Okay, so let's work it out. So how many times do we see the A allele? We see it 3 times in 5 individuals, 1 time in 6 individuals, and 0 times in 7 individuals. So we see it 3 times in 5 individuals, plus 1 time in 6 individuals, plus 0 times in 7 individuals. And then we divide that by the total number of alleles that exist in this population. And so let's do that really quickly. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. I didn't have to count that. So we have 9 times 18, which should be 81 times 2, which is 162 total alleles. Was that correct? Hopefully. 162. And someone thankfully did the math. Thank you, Casey. 0.389 is the allele frequency for A.
Actually, for my denominator, how did you get 162? Because I did 18 times 3. So I did something wrong, I think, because I multiplied total individuals, which is 18, by 3. And I multiplied by 3 because it's triploid. Is that wrong?
Oh, no, actually, you're right. Yes, because there is, wait, it should be 3 times 18. Yeah, because you don't do 1, 2, 3, 4, 5, 6, 7, 8, 9.
Yeah, I got 54 as my denominator.
1, 2, and 3. Yeah, sorry about that.
Okay.
Let's back it up. Oops. So not 162, not 9 times 18, but instead, this is not tetraploid. Let's revisit this because this is very important. We don't want to get lost. A triploid individual has 3 alleles representing its gene at any particular locus. Okay, no problem. Yeah, sorry. So not 9 times 18, but 3 times 18. And that gives us 36 plus 18, which is like 54 or something. Yes, thank you. Quick maths. And that would be 0.389. I was about to say. Okay, so moving on to A, the lowercase a. We see A 0 times among 5 individuals, 2 times among 6 individuals, and 3 times among 7 individuals. 0 times 5 plus 2 times 6 plus 3 times 7, all divided by the total number of alleles in that population. No, Maya, you cannot. So Maya asked if we can simply subtract this number from 1 to get the allele frequency of lowercase a. And the reason we can't do that is because, can anyone say?
Because
Well,
this is taking into account all the alleles, which there's 54. Is that right? 54
yes,
alleles?
exactly.
As
And
opposed to just like 2 A's.
yes, and in this 54, you find all of the B's and C alleles. So the reason we can't do a simple 1 minus 0.389 equals the lowercase a is because the uppercase a plus the lowercase a is not the total number. These do not comprise every allele within this population because there's also capital B, lowercase b, and capital C. So 1 should equal A plus B plus B plus C. Yeah, you can do it for the last one once you've found out all the others. Dun, dun, dun. And usually, people have mentioned Hardy-Weinberg in the chat. Usually you end up doing the 1 minus one of the alleles equals the other allele frequency because in Hardy-Weinberg, we're only looking at a single diploid locus where the only alleles that we're studying in this population are these two. So obviously, the total number of alleles, 1.0 or 100%, is going to equal the frequency of this allele plus the frequency of this allele in the population, because these are the only two alleles that make up this 100%. So this is some math. Did someone do that math? Nope. Anyway, we can do the math later. So capital B, what's capital B? How many times do we see capital B in the 5 individuals? Yes, 3. So we see the capital B 3 times in 5 individuals, like Caroline said. Someone said 0.661. Wait, no, that'd be 0.389 or 1 minus 0.389. Is that right? Interesting. We can explore that later. Huh.
No, Garrett, I think it's okay because we're not dealing with more than one different type of allele for A. So if like the first question that we went over, if we were doing like A1, A2, and little a, because we have 3 different alleles located at that locus, then we wouldn't be able to do 1 minus. But because we only have 2 different alleles at that locus, we can still do 1 minus, whatever the other one is.
Actually, the reason that we're getting 0.389 and 0.611 is because this 54, this 3 times 18, this is only considering the A locus for all these individuals. And I don't think that's correct. I think we should be considering the 9 times 18. Okay, the answer key says it's correct, I guess. That's not how I'd be doing this, but I guess I'd be wrong then. So moving on.
No, Garrett, it would still be 3 times 18 because you're only looking at the A group, like the number of A alleles that you want. Or like the frequency of the A alleles. So when you're finding the frequency of each allele, you have to look at them just like on their own. So like only look at the A's and only look at the B's. And
So
the
when you do
C's.
allele frequencies, you only look within a locus. Okay, that makes sense, I guess. Yeah. So I guess this is only asking frequencies of each allele within a locus. There are some letters being thrown around in the chat that I don't understand. Yeah, C would just be 1 then, because the only kind of C allele is capital C. Yes, Joseph. For quiz 7, I haven't seen quiz 7, I'm sorry. Anyway, yeah, we can keep doing this math, and this is the math you do if you want to find the frequency of any allele at any particular locus. You simply find the number of times you see that allele in or among all of the individuals of the population. So we find the number in the denominator by multiplying 18 by the number of alleles within a locus. Yes, because there are 18 total. Yeah, because there are 18 total, and then there are 3 alleles that make up, or yeah, multiply by the ploidy. Exactly. So yeah, I'll spare you the rest of the math. Let's take coupling and repulsion. No, we have not gotten to cis and trans recombination.
Sorry to interrupt, Garrett, but Usman, I just looked at quiz 7 and there's a tiny bit of math, but it's not anything like what we were just doing. Like it's not like super long, complicated math.
All right, so that was quiz 6. It was tough for a lot of us. It would have been tough for me, apparently. Is there recombination questions? There will be some recombination questions. We did go over very lightly what recombination is, recombination versus parental genotypes. But we didn't go too much further into depth than that. And if you wanted to see an example, you can look at question 5 on the week 7 review. And in it, it simply asks, what percentage of offspring are recombinant based on a cross that we see? Did you want to explore that some more, Maya? Okay. So let's see if there's a nice example waiting for us. Certainly, Carla. There's a lot of Hardy-Weinberg, a lot of genotype and allele frequency calculation.
Basically, yeah, it'll be pretty math heavy.
Just algebra, though. Just try to keep clear all of what a question is asking for. It shouldn't take the whole time. It's quick maths. um, if you consider the extra credit questions, then kind of. Speaking of, I think we've articulated it before, but if you want to find the answers to the extra credit questions, no matter what question it is, for this exam at least, you will find those answers in the About Your Instructor and Assistants tab. So read and learn about us.
Joseph, yes, there was a secret question today, but we'll say it again one more time at the end before we end. Carl, are you asking if there are 1 or 2 extra credit questions? How many were there last time on the exam? I can't remember.
3,
Because I think there were 2 last time and we had given out like 3 questions. So probably like 2 again this time, I would assume. 3 maximum.
Oh, we haven't gotten to 3-point crosses yet.
No. I think someone at the very beginning asked if, oh, Usman, do you still want to go over slide 46 from chapter 25? Yes, okay. I have, look at that, I have the chapter 25.
Yeah, it was
Slide.
the concept question number 7, I believe. Yes, concept check 7. Sorry.
Let me, okay, can you all see my screen?
Yes.
Okay, so the question is asking, in each generation, 10 random individuals migrate from population A to population B. What will happen to the allelic frequency Q as a result of migration when Q is equal in populations A and B? All right, so
Yeah, the answer they gave us, it was C, but I'm very confused. I don't know why, because it looks like it will increase because it's going from A to B. So it will decrease from A and it will increase in B. But I don't know why it's not changing. It just says it's not going to change.
I think the reason why it doesn't change is because you're taking, well, at the beginning, we know that Q is an equal frequency. Or yeah, so
because
Q
you're
is the same. Q is the same in A and B when we're starting, and we're just taking 10 random individuals. So I think the fact that it's random, we're not like cherry picking for Q is the reason why it doesn't change.
Oh, I
think it's-
see.
I think it's because the amount of Q doesn't change. It's just in a different place. So the frequency overall of Q is accounting for the same, 2 populations of A and B. So it would be the same amount of Q. So, nothing is increasing, nothing is decreasing, just Q is moving. So overall, it's still going to be the same amount of population.
Yeah, Maya, sorry to interrupt. Maya and Joseph have it right on the nose. Like, because it's random, just as many Q individuals or just as many Q alleles are moving forward and backward, or just as many Q alleles are migrating from population A to population B as P alleles. So the frequencies shouldn't change.
I guess it's because of the equilibrium, right? Because of the forward and reverse mutation, I guess, right?
Well, technically, there isn't migration from population B back to population A, as the question states, but just as many, because it's random, we can assume that just as many P alleles are leaving population A as Q alleles leaving population A. And so the numbers change, but the ratios don't.
Okay, got it. Thank you.
You're welcome. Mutations or migration question mark. Forward migration equaling reverse migration. Did I misspeak? Maybe is that what you're?
I was asking because the question states random individuals migrate from population A to population B. So we're talking about migration rate because you said somebody hit it on the nose. I don't remember the name of the person who hit on the nose, but I just like basically wrote down their answer so that I could refer to it later, but their answer had said mutation. I just wanted to clarify that the answer, we're talking about migration, not mutation, right?
We are talking about migration. I didn't read their
Okay.
whole
No,
comment.
that's fine. It
Read
starts with
the-
the M at all. I just wanted to check because I'm going to look at this later. Thank you.
Sorry, yeah, I just read the like forward equaling reverse, but it is definitely migration as the question states. And mutation, that is a black book that we can open if you want, but you like really have to pay attention to the wording so you don't confuse it. Okay. I mean, it is possible that like you see it all the time in plants, animals, living organisms, that there can be some mutation that causes a population to evolve. And then in the descendants of that evolved population, that derived population, can mutate exactly backward to the original condition. That's possible. Exactly, everything's random. Can we go over the practice problem in slide 27 of chapter 5?
Yes. Let me pull up that PowerPoint.
Oh, I remember the one, blood types and RH factor. That's like the B's and positives and negatives.
All right. Let me stop sharing this PowerPoint and then start sharing the other one. Actually, something that I did want to mention while I'm pulling up PowerPoint. Someone asked if there were like you could have complications with a mother and a baby having different blood types, and like this won't be on your exam, but just something that like I thought you guys might be interested in if like the person who asked about it is here. The only time that you'd have a complication between a mother and the fetus regarding blood types is if a mother is Rh negative, so that means that she has a negative blood type, and her baby is Rh positive, which means it has a positive blood type, and this is her first pregnancy. Then if she has a second baby and the second baby is Rh positive as well, then her antibodies can attack the second baby's blood and like result in a bunch of complications, but that won't be on your exam. I just thought it would be something interesting to throw in. Okay, so we want to look at slide 27. This one. Oh, wait, I'm in chapter 4. That's my bad. I was really confused. I was like, this does not mention RH. Okay, I have the correct one pulled up now. So this question, right? Okay, so a woman who is blood type A, Rh negative, marries a man who is blood type B, Rh positive. The husband and wife each have one parent who has O negative blood. What are the genotypes of husband and wife with respect to blood type and RH factor? Okay, there's a lot to unpack here. The first thing that I would do is try and find the blood types of the woman and the man. And so the woman we know is A, so she has to have at least one A allele, and then we're just gonna ignore the second one for a second. And then she's RH negative. So she's RH negative. Can anyone tell me her genotype? Anybody? Yeah, so she'd be RH minus, RH minus. And so this is woman, I should probably label that. All right, and then man is going to be, yeah, so it'd be RH negative because it's a recessive genotype. And so the man is going to be, he has to have at least one B. And then because he's positive, exactly as Nicholas said, it'd be RH plus, and then RH, we don't know the second one, because positive is dominant. And yes, Joseph, you are one step ahead of me as always. So because because the woman and the man both had a parent who had O negative blood, that means the O is telling us that both of our individuals are going to have one O allele. I feel like I should have done better notation because traditionally it's like IA and then little I. And so I guess we'll stick with the traditional notation. All right, so because each one had a parent with O negative, they're each gonna have one allele that represents O. And Carla, yeah, basically you have to know that RH negative is recessive. But if you encounter this on the exam, now you will know that RH negative is recessive. And since the parent was O negative, that negative is telling us that the parent was RH negative. So that means that the man's second allele at the RH locus is going to be RH minus. And then that is all that it's asking us. So does it make more sense this time around for everyone who found it confusing before? We have one yes. I'll also write in the parent's genotype here so it's you can see it.
When the question says that the parents are O negative blood, does that imply that the parents are RH minus? It looks like yes, because that's how it's written.
Yes. So when they're,
sorry, go ahead.
oh, no, you go.
So RH negative means negative blood type. RH positive means positive blood type. Like
Wait,
that's
sorry.
like the RH factor, if it says RH negative, that is telling us that it's a negative blood type, and RH positive is telling us it's positive blood type.
Mm-hmm.
Got it. Okay, so RH factor is saying if it's positive or negative blood type.
Yep.
Okay, so that's how we know the parents are O negative. Okay, that makes sense. Thank you.
Yep, Joseph got it right. Exactly. RH minus means negative. RH plus, RH minus, or RH plus means positive. Does anybody else have any questions about that? Is there anything else that anyone would like to go over? Any other questions or anything from the slides or concepts we could go over? Other quiz questions also from previous quizzes if you guys had questions about those.
I will ask a question.
Mhm.
So slide. Okay, 3 different questions. I don't know if bar bodies are going to be on this test, but if they are, maybe we can go over chapter 4, slide 47.
Yes, absolutely.
And I talk about it in the video that I uploaded earlier.
Okay, cool. I'll have to check that out.
Let me see. This one, right?
Yes.
All right, so how many bar bodies does a male with XXXYY chromosomes have in each of his cells? So what do you guys think?
And they can see the answer.
Oh, that's awkward. Wait. Okay. Oh gosh, you can still see it at the bottom. Okay. Well, you guys have already seen the answer, but it's
So will you
2.
tell me why?
And yes, someone do tell us why. Yes, just while you guys are explaining why, I will make up another question.
Maybe we could also talk about what bar bodies are, because that was never super clear ever since we did that case study on the Komodo dragons. I never really got a complete understanding on
Yeah,
what they
absolutely.
are.
Sam, can I share my screen?
Oh yeah, of course.
Okay. I got kicked out of Zoom apparently. Get out of here. Oh, God damn it. Okay. Dun dun dun. I should have had it prepared. Sorry. Okay, so bar bodies. Where are you at? Okay, so let's make this bigger. So a bar body, before we talk about bar bodies, we know that, for example, in mammals, like this cat, the homogametic species is what kind of individual? Yes, XX. And what kind of individuals have XX chromosome? Females, yes. So females are the homogametic members of most mammal species because they have 2 of the same kinds of gamete, 2 of the same kinds of chromosome. And so in every kind of cell that is in a female mammal's body, let's say that this is a human's female's cell. In this cell, it is going to have the genotype XX. And this is okay, I guess, but do we want both of these X's running around in our cell? You might think like, oh, I have 2 X chromosomes. I can make all the stuff that I want. But that is not really the good stuff. So on these X chromosomes, obviously there are instructions. Let's say there's instructions for a kind of cytotoxin. And one X chromosome produces just the right amount that kills the bad stuff and doesn't do anything else. But if you have 2 that is also creating just as much cytotoxin as the 1, then you have twice as much bad stuff in your cell, twice as much product that is being used for a destructive purpose. Exactly, there's too much instruction, too much stuff is being made. And so this is a problem. We're overdosing on this cytotoxin that we're making. Obviously, not all cells make cytotoxins. Some kinds of cells make, good stuff, make energy, but you don't want too much energy. because chemistry happens in a cell. Like, for example, in a plant cell, you don't want too much rubisco running around doing the Calvin cycle, because then if you have too much oxygen, I'm not going to get into the chemistry, but we don't want to overdose on the products that are made by the instructions of a single X chromosome. So we need to reduce our dosage. And so what happens is a process called dosage compensation, which is where one of these X chromosomes gets deactivated. And when it's deactivated, it gets turned from this nice little X shape, gets condensed down into a little blob. And that's what this bar body is. It is a condensed, deactivated X chromosome. This was discovered in the 20th century by a geneticist named Mary Lyon. And bar bodies are named after a Canadian scientist named Murray Barr. Kinga's cat saw the cat and they wanted to
Yeah.
contribute.
Yes, because I'm eating food and he's being naughty. Hi, everybody.
Kitty!
Bye.
No, bring the cat back.
I know, I'm trying to get him out of here.
So Casey, did that, do you have any questions about that? Or does anyone have any questions? You're right, we didn't talk too much about it in lecture.
That's really good explanation. But for concept checked 8, so basically it's asking how many bar bodies does a male with XXXYY chromosomes have in each of its cells, and they're saying 2 bar bodies because this individual has an extra X chromosome and an extra Y chromosome, because typically males are just XY, but this one is like XXXYY. Is that why it's 2?
Yes, so in males, typical males, you have XY, and it's the X chromosome that makes most of the good stuff that we need, or that our cells need. So males don't have to do dosage compensation because they only have one X. This Y, we don't really have to worry about it when we're talking about cell products. And so dosage compensation doesn't happen. You don't find bar bodies in male cells, assuming that they have a single X. You don't find that because they only have one X to begin with. So when we're looking at this individual, we can kind of disregard their Ys because their Ys aren't contributing to the products that might cause our cells to overdose. So once again, our cells only need the instructions that come from one X chromosome. So in this case, because they have 2 extra, these 2 will both be condensed down into bar bodies so that we maintain our single X dosage. Am I muted? Okay, no, good.
That makes sense. So Y chromosomes would never be bar bodies.
No.
Okay.
They're
Yeah,
weakened
that
when
makes
they
a
don't
lot
do
of
it.
sense now.
Yeah.
Thank you. So really, I mean, if we only need one X chromosome, then
Well, there's actually
that's not in
dosage.
every scenario, right?
Well, we can look at this cat and see that there's some benefit to dosage compensation. And so we see that this cat, the reason that it's partially orange, partially black, like at different individual cells create different kinds of, or inject different kinds of pigment into the hair follicles. And that's because this cell versus this cell versus this cell versus this cell versus this cell is going to have one or the other X chromosome deactivated. And we don't really understand how the cell decides whether this one or this one gets deactivated. But on the whole, because we don't know, and because there isn't like a clear tendency, we just assume it's random. And so there's kind of like a random distribution of orange hairs on this cat versus black hairs versus orange versus black. And obviously in this cat, their cells are coding for more than just pigment. So this cell over here could have the X chromosome active that has really cool anti-carcinogen chemicals, but maybe this X chromosome didn't. And maybe this cell over here had the not cool kind of carcinogen information. So this is called mosaicism because, they're like a mosaic. They have different cells coding for different things just because, even though they have the same information technically, the information that's being expressed is different because this chromosome is most likely going to have different alleles than this chromosome. It's called female mosaicism. And that's why calicos are female. Yeah. Or I guess it's not why calicos are female, but female calicos, or calicos are most often female because, well, that's just kind of because of, well, yeah, actually, you're right, sorry. But this theoretical genotype is also the reason why you can have male calicos. For example, this individual would be a male because they have a Y chromosome, and that would kind of like take over the process of their sexual development. And but the male would have access to 1, 2, 3 different X chromosomes, and it's random whether or not this one would get deactivated versus this one versus this one. So maybe this one coded for orange hair, and this one for black, and this one for white. And so at different cells, or different cells at different points in a male cat's body, a male who has this genotype would show that modeled calico appearance. But you'd obviously only see that in a male cat that has more than 1 X chromosome, which is very unusual because most males only have 1 X chromosome. So it is chosen by advantage. Actually, Nick, that is a very strong avenue of research. I think it's amazing. And if that were the case, it would be so crazy that you have like natural selection, not going on in a population, but within your own body, like different cells competing kind of based on whether or not significantly helpful traits are being activated or deactivated by dosage compensation. That is amazing to think about. How about another example?
Usman, example of bar bodies? Oh, yes. The one that I literally just made up. We could do that one. Or Garrett, do you want to make one?
Oh, you got it.
Okay. All right. So here is the one that I made up, the one that's highlighted. So how many bar bodies I spelled bar wrong. How many bar bodies in a female with XXXXX?
I don't know.
Not quite. There might be more than 3.
Maybe.
Yes. So there are 4 bar bodies. And so 4 bar bodies. So basically what happens when you have your X in activation, literally like for any individual across the board, you only need 1 X chromosome in each cell to like still be like a functioning normal human. So we didn't talk, I don't believe that we talked about this in class, but you can have an individual who's XO and they're still normal. I want to say this is Turner syndrome, but I am not entirely positive.
Yeah, we haven't gotten to aneuploidy yet, but I think that's later in the semester.
So Casey, we can, it is weird that we have two, even though we only need one, but I guess it's just the way that we have evolved.
Yeah, maybe female mosaicism is it's highly selectively advantageous. And that's why diploidy hasn't gone out of style. Because, it takes twice as much genetic information, twice as many molecules to be diploid as it does to be haploid. So,
And Caroline asks, so when we are counting the bar bodies, is it just the number of X's after the first one? Yes. So like quick shortcut way is just like you take the number of X's you have and subtract one, and then what you're left with is the number of bar bodies you'll have. Does anybody else have questions about bar bodies? Or any other?
Oh, sorry. I was answering Nick's question. Yes, pentaploids are a thing. You see that in plants a lot. Plants that have like non-disjunction events. Actually, there was even I think the first extra credit seminar was about polyploidy in plants. But the reason that pentaploids, like you don't find independent pentaploid individuals whose entire genomes are pentaploid, can anyone hazard a guess? Has something to do with a cell cycle? Bingo. Yeah, when you divide in anaphase, the spindle fibers typically split the homologous chromosomes in half, or like in half by number. Hopefully you can visualize that. Like if there were 4, it would drag 2 to one side and 2 to the other side. But when there's 5, how do you evenly separate 5 into 2 or to the 2 different poles of the cell? You can't. And so some funky stuff happens. And instead of getting haploid chromosomes, you get like 2n plus 3n from a 5n. And that just doesn't go well. Usually it's 3 decimal places after the answer. That's just what I noticed for all of the questions we've had so far, but I guess it's kind of arbitrary.
We do like a maybe more difficult from chapter 25, like the calculating of everything, like the genotype, phenotype, like all of that.
Of course. Do you want to maybe bring that up, Sam, please?
Yes. Do you have like a particular example that you'd like to go over, or?
Yeah, so I mean, I'm looking from the book. There is in number 18. It's from the book though. It's example 18.
Okay, I do not have a book in front of me right now.
I can bring one up really fast. Okay. Let me find it. Sorry, 1 second. Okay, here we go. Let me share my screen. So, a group of scientists are studying a gene that causes outy belly buttons. They have found 2 alleles involved in the belly button phenotype, one being capital and the other lowercase a. The scientists then genotyped, or found the genotypes of the 500 individuals and discovered this distribution of genotypes. So let's find the genotypic and allelic frequencies for this population. Where do we start?
Total. What's the
Yeah.
total
That
number?
is a good place to start. We find the total number of individuals in this population. 325 plus 75 is 400. Well, actually, I guess it does state that there's, 500 individuals. So now that we know the total number of individuals in the population, this is the total number of genotypes that we will see. Because, every individual has a genotype. And if there are 500 individuals, there's 500 genotypes. Obviously not 500 different genotypes, but 500 total genotypes. And so for the genotypic ratios, the genotypic ratio of the homozygous dominant is going to be the number of homozygous dominants divided by the total number of individuals, which, like someone just said, is 325 divided by 500. And then for AA, it's 100 divided by 500. And then the homozygous recessive is 75 divided by 500. And so those are the genotype frequencies for the individual genotypes: homozygous dominant, heterozygous, homozygous recessive. And yes, we can continue, just like Nick said, we can further what we were doing earlier. And in order to find the allelic frequencies, for example, if we wanted to find the allele frequency of capital A, of the capital A allele, the frequency of the capital A allele is just the number of times we see the capital A allele divided by the total number of alleles that exist in this population. So in these 325 individuals that are homozygous dominant, we see the capital A allele twice. So we have 2 times 325, because there are 325 individuals and they each have 2 capital A alleles. Then we can move on. In 100 individuals, we see that the capital A exists only once. So we have 2 times 325 plus 1 times 100 plus 0 times 75, because these homozygous recessive individuals don't have a capital A in their genotype. Someone said something in the chat. Yeah, okay. And so we have the frequency of capital A is going to be 2 times 325 plus 1 times 100 plus 0 times 75 divided by the total number of alleles in this population. Because remember, allele frequency is the frequency of a specific allele, or how often we see a specific allele for all of the alleles that exist in this population. You don't necessarily need to write the 0, no. Yeah, it just helps me to understand what's really, or what I'm really looking for. And so we have 650 plus 100, 750 divided by the total number of individuals times 2, because every individual has a genotype, and that genotype is comprised of 2 alleles. So 750 divided by 1,000 is 0.75. And that's the allele frequency for capital A. And because we only have 2 different kinds of alleles in this population, we know that the frequency of this capital A allele and this lowercase a allele are going to be all of the alleles in that population And therefore, the frequency of capital A plus the frequency of lowercase a is equal to 1. And because we just found the frequency of capital A, we can subtract it from 1 to very quickly find the frequency of lowercase a. Or you could do the same math, and we see that in these 325 homozygous dominant individuals, we see lowercase a 0 times. In the heterozygous individuals, we see lowercase a once. There are 100 of them. And in homozygous recessive, we see lowercase a twice. So we'd have 2 times 75 plus 1 times 100 plus 0 times 325 divided by 1,000 alleles total. I suppose you could be asked the frequency of a heterozygote, the genotype frequency, and that would be very easy. We just do 100 divided by 500. Yeah, the frequency of heterozygotes in that population is the number of heterozygotes divided by the number of individuals. Of course, yeah, and actually I think we're going to be cutting the review in just a few minutes. So let me pause the recording really fast.
Coming with questions.
Yeah, and we are meeting tomorrow for class. We'll be talking about stuff that's not on the exam. Absolutely, yeah. And please let us know what you think of this kind of review instead of us making a bunch of questions and spending the whole time on them. Yeah,
We're
thank you
glad to
guys
hear that
for-
it helped.
Oh, what'd you say?
Oh, no, I just said we're glad to hear that it helped because a lot of people were saying it was helpful in the chat. Nicholas says both types help. Having questions and just doing like open-ended stuff.
Okay, yeah. Oh,
All
yeah,
right, well,
good point. Thank you. If you have the time, I suggest going here, LA Resources, go to the bottom, and I made some videos going over most of the big topics that'll be on the exam. Night, Nick. Love you. But yeah, thank you everyone for coming, and I hope you all have a great night. We'll see you tomorrow.
all right, see you guys.
Bye-bye.